Hello, we are going to talk about replication and repair. It's the phase which occur before then cell division. Okay, the DNA will divide first before the cell occurs the cell division. Okay, so we can start with the DNA replication. Talk about the why occurs the replication. Because the genetic information must be transferred to the parent to progeny. Okay? The replication is a process in which two daughter DNA molecules are produced that are each identical to the parental DNA molecule. Two complementary strands of the parental DNA are pulled apart. Okay? Each of these parental strands is then used as a template for synthesis of a new complementary strand. During cell division, each daughter cell receives one of the two identical DNA molecules. Okay? You can see the sequence of that occur replication in prokaryotes. Okay, here you have circular DNA, and you have one single point to start the replication, and after termination, that two DNA sequence will be pulled apart in the cell division. Okay? So the bacterial chromosomes are closed, double-stranded circular molecules having a single origin of replication. Separation of two parental strands of DNA creates two replication forks. Replication forks move away each other in opposite direction around the circle. Replication is a bidirectional process. The two replication forks eventually meet, resulting in the production of two identical circular molecules. Origin of replication in eukaryotic chromosomes should be multiple, and each one having a process identical to the one single eukaryote. In the eukaryotic process, each chromosome contains one molecule of DNA. So in the DNA replication, it starts with the unwinding the DNA sequence, and after start the process of making a new strand. The DNA has multiple origins of replication. Bidirectional replication occurs by means of a pair of replication forks produced at each origin. Completion of the process results in the production of two identical linear molecules of double-stranded DNA. The DNA replication occurs in the nucleus during S phase of eukaryotic cell cycle. The two identical sister chromatids are separated from each other when the cell divides during mitosis. Steps of replication. The first one is made by helicase. This is a protein which, after recognition of the site of replication, that protein breaks the hydrogen bonds holding the base pairs together, form the replication forks. The single-stranded DNA binding protein is a molecule which protects from the degradation by nucleases and prevents them from the reassociation before the mitosis is completed. Before the duplication is completed. The second step, the primase, synthesizes a short RNA, about 10 nucleotides, which is called the RNA primer in the 5 to 3' direction. The primer begins at the origin of each parental strand, and above you have the leading strand, which has just one RNA primer, and the lagging strand below, which will have a multiple RNA primers, and each called Okazaki fragment. The parental strand is used as the template for this process. Okay, so this is the parental strand, separated by the helicase, and the DNA binding, single binding protein maintain this separation, and the RNA prime gives it at the beginning of the replication, and this is parental strand, and this is in blue is a parental strand, and in red a new synthesized strand. Okay. RNA polymerases are required because DNA polymerases are unable to initiate synthesis of DNA and can only extend a strand from the 3' end of a preformed primer. Okay? This primer is formed from 5 to 3', and the polymerase can read after 3' and to form a new DNA. So this is a resume from this. You have a helicase to separate the two strands. You have a single strand embedding binding DNA protein to protect and avoid the binding the DNA, and you have the polymerase to read and making a new DNA polymerase, new DNA strand. Okay. The third step the DNA polymerase III begins synthesizing DNA in the 5 to 3' direction, beginning at the 3 ends of each RNA primer. The newly synthesized strand is complementary and antiparallel to parental strand used as a template. This strand can be made in one long piece and is known as the leading strand. So, the leading strand is it will have one single RNA prime. In the lagging strand is synthesized discontinuous as series of small fragments, about 100 nucleotides long, known as Okazaki fragments. And the lagging strand must have multiple DNA primers and multiple sites of start replication. There is a leading and a lagging strand for each of the 2 replication parts on the chromosome. Okay? So the last RNA primer, like here, is removed by RNase age in eukaryotes. An uncharacterized DNA polymerase fills in the gap with DNA. The gap in the DNA. In prokaryotes, DNA polymerase 1 both remove the primer in the 5' exonuclease and synthesizes a new DNA, beginning at the 3' end of the neighboring Okazaki fragments. DNA ligase seals the mix between Okazaki fragments, converting them to a continuous fragment. We can see here a proofread properties of DNA site synthesis. So both eukaryotic and prokaryotic DNA polymerases have ability to proofread this work by means a 3 to 5' direction exonuclease activity. And you can see here a mistake about guanine with a cytosine, and this polymerase making unpaired base, and we can have a 3' and exonuclease to left this guanine and make a correct part of base. Okay. DNA gyrase or DNA topoisomerase provides a swivel in front of each replication force. So, swivel, and the helicase can unwind. Helicase unwinds the DNA at the replication force. The DNA ahead of it becomes overwinding, and a positive supercoils form. DNA gyrase inserts negative supercoils by making both strands of DNA passing the DNA strand through the nick, and then resealing both strands. Okay. DNA polymerase have different types, and each one has one function. The DNA polymerase alpha and delta work together to synthesize both the leading continuous and the lagging discontinuous respectively. Alpha leading strand and the delta lagging strand. DNA polymerase gamma replication mitochondrial DNA. Remember that? Because there are a lot of questions in tests ask about what is the function of each one. And if you remember, gamma just occurs in mitochondrial DNA. DNA polymerase beta and epsilon are thought to participate primarily in DNA repair. DNA polymerase epsilon may substitute polymerase delta in certain cases. Okay? Telomeres. Telomeres are repetitive sequences at the end of linear molecules in eukaryotic chromosomes. With each round of replication, in most normal cells, cannot complete synthesis of 5' ends of each strand. This contributes to aging of cell, because eventually the telomeres become so short that the chromosome cannot function properly and the cell dies. Cancer cells have relatively high levels of telomerase to synthesis of new telomeres. Cancer cells thus prevent the telomeres from becoming shortened and contributing to the immortality of malignant cells. So, we can have a resume about the protein involved in DNA replication. So, the first step is recognition of origin of replication. It's making by DNA a protein in prokaryotic cells and is unknown in eukaryotic cells. Unwinding of DNA double helix occurs by action of helicase in both prokaryotic and eukaryotic cells. Stabilization of unwound single-stranded DNA binding protein occurs by synthesis of RNA primers is making by primase in both eukaryotic and prokaryotic. Synthesis of DNA of leading strand in DNA polymerase 3 in prokaryotic cells and leading and lagging strand. For eukaryotic cells, the leading strand occurs by DNA polymerase gamma, and the lagging strand occurs by DNA polymerase alpha. The leading strand polymerase omega. Removal of RNA primers in the 5' to 3' exonuclease occurs by polymerase 1 in prokaryotic and unknown in eukaryotic cells. Replacement of RNA with DNA occurs by DNA polymerase 1 in prokaryotic and is unknown in eukaryotic cells. Joining the Okazaki fragments occurs by DNA ligase in both eukaryotic and prokaryotic cells. Removal of positive supercoils ahead of advancing replication forks occurs by topoisomerase 2 known to DNA gyrase in prokaryotes and DNA topoisomerase 2. The other important enzyme is a reverse transcriptase. It's an RNA-dependent DNA polymerase. So you make DNA from RNA. Requires an RNA template to direct the synthesis of the new DNA. Retrovirus, most notable HIV, uses this enzyme to replicate their RNA genome. DNA synthesis by reverse transcriptase in retrovirus can be inhibited by AZT, BBC, and BBI. The reverse transcriptase activity is associated with the telomeres in normal eukaryotic cells, and it's encoded by a retrotransposomes residual viral genomes permanently maintaining in human DNA that play a role in amplifying certain repetitive sequences in DNA. Okay, this is now a new topic, the DNA repair. The structure of DNA can be damaged in a number of ways through exposure to chemicals or radiation. Correct bases can be incorporated during replication. Any defect in DNA repair carries an increased risk of cancer or mutations. Most DNA repair occurs in the G1 phase of eukaryotic cell cycle. Mismatch repair occurs in the G2 phase to correct replication errors. Timing timers. You can see this picture about the one timing in yellow and this timing form a timer. It's caused because the UV radiation, the recognition excision enzyme, excision endonuclease, repair enzymes, DNA polymerase and DNA ligase. The disease most common occurs timing dimers is scleroderma pigmentosum. It's occurred by extreme UV sensitivity, excessive freckling, cluster of concentrated melanized cells, multiple skin cancers, and corneal ulceration. The other error in DNA replication occurs a mismatched base. Cause a DNA replication errors. Recognition excision enzyme, mutation on one of the two genes, HMSH2 or HMLH1, initiates defect repair of the DNA mismatches. Non-polyposis colorectal cancer is the most common disease associated with mismatch repair. You can see here a molecule of the cytosine, and if you deaminate the cytosine, you make an uracil, okay? Called spontaneous or by HET. Loss of amino groups from cytosine converts cytosine to uracil. Recognition excision enzyme uracil glycosylase recognize and remove the damaged sequence. Subsequently, is recognized by AP endonuclease. Repair enzyme DNA polymerase fills in the gap, and the DNA ligase nick in the required strands. Okay? So an example of keratoderma pigmentosus. It's an autosomal recessive disorders which occur 1 to 250,000 per nascimento, born, extremely sensitive to sunlight, screen flattening, ulceration, and skin cancer. Most common deficiency occurs in the excision endonuclease. Diagnosis measurement of the relevant enzyme excision endonuclease. The hereditary polyposis colorectal cancers, also called Lynch syndrome, due to autosomal dominant mutation of DNA nucleotide mismatch repair gene that predisposes for colon rectal cancer, associated with increased risk of endometrial and ovarian carcinoma. The gene mutation, or usually age MLH1 or age LH2, that encodes enzymes that carry out DNA mismatch and repair. In families with carcinoma polyposis, intestinal individuals may inherit one non-functional or deleted genes. This is manifested in intestinal cells because they are constantly undergoing cell division. Some questions to recall. What are the major types of DNA damage repaired by each of the following pathways? Photoreversal, base excision repair, nucleotide excision repair, and mismatch repair. For photoreversal, pyrimidine dimers are only type of damage repaired by photoreversal. The base excision repair, damaged or missing bases. The nucleotide excision repair, pyrimidine dimers, and other damage that leads to distortion of the DNA structure. And the mismatch repair, replication errors. Other question, specify which of the repair pathways listed in the previous question is associated with each of the following enzymes protein complexes or genes produced: CSB, AP endonuclease, HNSH2 DNA photolyase, ERCC6, MUC, XPA, uracil DNA glycosylase, DNA ligase RAB3, EF2H, DNA polymerase. So, pyrimidine dimers, photolyase, damaged or missing bases, AP endonuclease, uracil DNA glycosylase, DNA ligase, DNA polymerase. Pyrimidine dimers, CSB, ERCC6, XPA, RAB3, EFLLH, DNA ligase, DNA polymerase. Replication errors, HMSH2, MUC, DNA ligase, DNA polymerase. Okay? Naming two human diseases caused by defects in different DNA repair pathways. That is, one of the two diseases you name should be in one repair pathway and the other disease should be in a different pathway. List briefly the steps in each pathway and then in more detail describe the normal function in that pathway of the protein encoded by the gene, which then, when muted, can cause disease. So, DNA repair, hereditary non-polyposis colon cancer, which is associated with a mismatch repair. Keratoderma pigmentosum, Coxsackie syndrome, and trichotillodystrophy, which are associated with nucleotide excision repair. Of these, the first two is the best to answer, although CS and TTD involve defects in the protein required for nerve. These proteins are also required for transcription. In CS and TTD, the mutation affects primary transcription rather than repair. The question asked for accompanied by symptoms of Keratoderma pigmentosum, and the defects are in primary in transcription pathways, not repair pathways. The other question is now believing that the substantial proportion of the single nucleotide substitution causing human genetic disease are due to misincorporation of bases during DNA replication, which proofreading activity is critical in determining the accuracy of nuclear DNA replication and thus the base substitution mutation rate in human chromosome. So the answer is 3 to 5 prime exonuclease activity of DNA polymerase delta. Explanation, you can see the DNA polymerase gamma is mitochondrial activity, and DNA polymerase 3 in prokaryotic cells do not participate in this process. The proliferation of cytotoxic T cells is marked in part upon infection with a newly discovered human immunodeficient virus designed HIV-5. The defect has been traced to the expression of a viral encoded enzyme that inactivates a host cell's nuclear proteins required for DNA replication, which protein is a potential substrate for the viral enzyme. The answer is single-stranded binding protein. The other one is not related to replication. The other question, the deficiency of an excision endonuclease may produce an exquisite sensitivity to ultraviolet radiation in Keratoderma pigmentosum. Which of the following functions would be absent in a patient deficient in this endonuclease? So Keloderma pigmentosus is a famous by removal repairing the pyrimidine dimers. The anti-pseudomonas actions of norfloxacin is related to its ability to inhibit chromosome duplication in rapid dividing cells. Each of the following enzymes participating in the bacterial DNA replication and is directly inhibited by these antibiotics. Norfloxacin is related to inhibit a topoisomerase 2. It inhibits the replication by inhibiting the unwinding the DNA. Cytosine adenosine C, RRC, is used as an effective chemotherapeutic agent for cancer, although resistance to this drug may eventually develop. In certain cases, resistance is related to an increase in the enzyme cytidine deaminase in tumor cells. This enzyme would inactivate RSC to form uracil adenosine. This is cytosine deamination resulting in uracil adenosine. So this keratosis congenital occurs in patients which have deficient synthesis of telomeres, which is unstable DNA resulting in cell death. Okay? Thank you. Oh no, more one. Single-stranded breaks in DNA. So, formation of the phosphodiester bond in DNA repair, not in the new DNA synthesis, is a DNA ligase. Okay? DNA ligase acts in the form of new DNA in the correction of the strand. Okay? So it's all for today. Thank you very much.